Welcome back. Today we're going to be talking about skeletal muscle physiology. And if you remember, physiology is just a description of how something works. So for example, with skeletal muscles, how do they do their job? How do they contract? So I've drawn out here a skeletal muscle fiber. And remember that a muscle fiber is the same thing as a muscle cell. It looks a little bit different than maybe other cells that you've seen that are big and round and are just big circles, but it has all of the normal things that a regular cell has, plus some special stuff. So a skeletal muscle fiber is kind of long and thin, and its job is to contract. So it's going to get shorter when it contracts. So this end will come in and this end will come in. And that is how the muscles get shorter, and then they exert force to pull your bones towards each other. Now, muscle fibers can't contract on their own. They require a signal from the nervous system. And so I'm going to draw in the terminal end of a neuron coming in and meeting up with our muscle fiber. Now, you might notice that they don't meet. There's a space between them. That's called the synapse. And so when we have a signal that comes from the nervous system and has to excite and get this muscle fiber to contract, it actually has to cross over the synapse from over here near the neuron to over here near the muscle. And this area right here where the nervous system and the muscle system meet is called the neuromuscular junction. A lot of people just write NMJ, neuro for nervous system, muscular for muscle, and junction for the place where that meets. And that's the terminal end of the neuron and the motor end plate of the muscle fiber. So a quick review of neurons. The kind of neurons that we've seen so far kind of look like this. There's dendrites at this end that receive signals. There's the cell body. There's a long axon. And then there's these sort of wispy terminal ends. And what we're looking at right here is one of these little terminal ends. And in fact, they don't exist by themselves, and muscle fibers also don't exist by themselves. When you have a muscle fiber, you are likely to have many muscle fibers, because they're all going to contract together, because each one of these is really small and unable to contract and move your bones by itself. So what happens is we have lots of muscle fibers that are together. A bundle of them is called a fascicle. And we also have a bunch of terminal ends. So you can see in the case of this neuron, we have 1, 2, 3, 4 terminal ends and 1, 2, 3, 4 muscle fibers. Now, in your body, you're not going to have such a small number of muscle fibers that are all associated with one another. You might have thousands or tens of thousands of muscle fibers that are all going to be stimulated by the same neuron. So we call everything that I've drawn on the screen so far a motor unit. A motor unit is one neuron and all of the associated muscle fibers that go with it. And so all we have to do is we have to send one signal down this motor neuron, and as it splits into all of its little terminal ends, every single muscle cell that's associated with it, that is being touched by it, that has a neuromuscular junction, is going to get the same signal at the same time. So all 4 of these guys are going to contract together when this one neuron sends the signal. And in that way, in the big motor units of your legs, you can have one neuron firing and sending the signal to contract, and all thousands of muscle fiber cells will contract at the same time. Okay, so we're going to close in a little bit on this one so we can see the synapse a little bit better. And on the motor end plate, which is this sort of thicker line over here, there's actually some special receptors that stick up. And these receptors are specialized to bind to the chemicals that are stored inside the terminal end of the neuron. So those chemicals are called neurotransmitters. They're called neurotransmitters because they transmit the signal from the nervous system to something else, maybe another part of the nervous system. In this case, we're transmitting the signal to a muscle fiber. So inside the terminal end are these little vesicles, and these vesicles are filled with a chemical called a neurotransmitter. In muscle contraction, we use a specific neurotransmitter called acetylcholine. Acetylcholine is often written as ACH, big I, big C, little h. So acetylcholine is the neurotransmitter that's stored in this particular terminal end of this particular neuron. There's lots of other neurotransmitters out there. You might have heard of a few like serotonin or dopamine. There's a couple other things that we have to set up so that once we do receive the stimulus, we can allow this to happen. So one thing to know is that we have a couple of gradients. We have a lot of calcium outside these cells. So it's not inside the cell, it's outside the cell, floating around outside near the synapse. We also have a lot of sodium. And we're going to use the fact that we have these gradients to help us get the signal from the nervous system to the muscular system, and also get the signal that we need deep inside this muscle cell and encourage it to contract. So what happens is you have a special signal that comes down the neuron. It's called an action potential. That action potential is really just an electrical signal, and we're going to learn more about action potentials in the next chapter. But for now, just think of it as like electricity coming down a wire, just like electricity travels through wires in your house. As the action potential arrives at the terminal end, it's going to trigger some changes in this plasma membrane of the terminal end. Remember, this is all plasma membranes. And so when that electrical signal comes, it's actually going to open special gates in the plasma membrane that are specialized for calcium. Now, when the gates open, what do you think is going to happen to calcium? There's a gradient. It's at high concentration out here and very low concentration inside. And so calcium is going to move down its gradient and move into the terminal end of the neuron. That's going to change some stuff here in the terminal end. All those positively charged ions moving into the terminal end are going to cause some changes, and they're going to cause these vesicles that are full of neurotransmitter to start to migrate over to the plasma membrane. So these are going to start coming over here and moving closer to the synapse. Now they still have their neurotransmitter inside, but what happens is once this piece of plasma membrane bumps up against this piece of plasma membrane, they're going to merge together. What you see is that this vesicle merges with the plasma membrane and releases its contents into the synapse. So we have a lot of acetylcholine that is in this area of the synapse and very little over there. Now, this is called exocytosis. It's almost like this terminal end of this neuron is just sort of spitting out the acetylcholine. And the acetylcholine will start to diffuse across the synapse. There's no membrane here holding it back anymore once we've fused together. So acetylcholine will start to diffuse across the membrane and will eventually bind to the acetylcholine receptors on the motor end plate. When that happens, these receptors will trigger gates to open in the plasma membrane of the muscle fiber. And these are sodium gates. So those sodium gates are going to open up. What do you think will happen to sodium? It's at high concentration outside of the cell and low concentration inside of the cell. It's going to go down its gradient and enter the cell by facilitated diffusion. As sodium floods into the cell, it's bringing a lot of positive charges into the cell, which is going to start another electrical event called an action potential on the sarcolemma of the muscle fiber. So far we've had an action potential, an electrical event on the neuron. We've released our neurotransmitter, which transmits signals from the neuron to the muscle fiber. And now we have created an action potential on the muscle fiber. This action potential travels along the sarcolemma. And remember, the sarcolemma is the plasma membrane of a muscle fiber. It is still made out of phospholipids, and we still have to obey the rules of transport. So the action potential travels along the sarcolemma until it reaches the T-tubule. The T-tubule is a specialized structure where we have an invagination of the plasma membrane into an area that's really deep inside the muscle fiber. So that action potential is going to travel down into the T-tubule, that electrical event. As the action potential moves down the T-tubule, it causes voltage changes to a structure called the sarcoplasmic reticulum. The sarcoplasmic reticulum lays on either side of that T-tubule. It's sort of a meshy structure. In a muscle fiber, the sarcoplasmic reticulum stores intracellular calcium, which is required for muscle contraction. So there's very little calcium inside the cell, but a lot of calcium stored inside the sarcoplasmic reticulum, or SR. So when that action potential comes down the T-tubule, it's going to cause some voltage changes, which will open calcium gates on the SR. So remember, I told you earlier that calcium and sodium are at very low concentrations inside a muscle fiber normally. When the muscle fiber is resting, you don't find a lot of sodium or calcium in the sarcoplasm. Remember that the sarcoplasm is sort of the goo that's inside of a muscle fiber. So as the calcium gates open, calcium is going to go down its gradient. So remember, calcium can't cross the membrane by itself because it's charged. And so it needs to go through a little gate or tunnel with facilitated diffusion. So calcium is going to go down its gradient from its level of high concentration in the SR to a level of low concentration in the sarcoplasm. And as calcium enters the sarcoplasm and the concentration starts to increase, that is what's going to trigger muscle contraction. You have a lot of calcium inside the cell, you're going to get contraction. When you have no calcium inside this muscle fiber, you're going to get no contraction. And so calcium is kind of a regulatory ion for skeletal muscle contraction. We're going to learn more about how calcium interacts with actin and myosin in the next video. But for now, let's just talk about all the different types of transport that we see here. The first one we saw up here was calcium moving down its gradient through a channel into the neuron. That's facilitated diffusion. The second one we saw is when the vesicles merged with the terminal end of the neuron, and we had exocytosis, which is bulk transport of acetylcholine out into the synapse. And then we had, that's number 2, sorry, forgot to write that. Number 3 would be diffusion. As acetylcholine moves across the synapse from an area of high concentration to an area of low concentration, we see diffusion. Once it binds to the receptors and opens the gates, we see another type of facilitated diffusion as sodium ions move through their associated channels into the cell. We're not going to really talk about what's going on with the action potential because that's for next week. But the last one we need to talk about is as the action potential hits up against the SR, the sarcoplasmic reticulum on both sides of the T-tubule, we're going to open those channels in the SR and allow the calcium to move out down its gradient. So that's another example, number 4 here, of movement or transport of materials across membranes. And this one is also facilitated diffusion. Come back again. We'll talk about actin and myosin next. See you in class.